Hey everyone, this is Brenda Pacheco and I'm doing this exercise for Bio 225 week number 3. This is exercise number 1 and I will be doing the direct and negative stains. So what I went ahead and did is I already grabbed one of my slides and I labeled it 1 direct M. I put the circle in the middle. So then it's telling us to scrape the inside of your teeth and gums with sterile swab. So I will be grabbing this sterile swab and scraping the inside of my gums. Okay, and I will be putting it in the circle that I did here in the middle of the slide. And then it says to allow the slide to completely air dry. So I will check back to do the next step. Okay, so now I have allowed my slide to air dry and I am grabbing the slide with the grabbers here. I forgot what they're called, but I'm going to pass this through the flame 5 times. So here we go. 1, 2, 3, 4, and 5. So I will go ahead and have my slide cool for 10 minutes and I will check back. Okay, so I have now let my slide cool for 10 minutes and now it says to apply some of this Crystal Violet Gram Stain, and I'm going to add about 1 or 2 drops to the slide and then wait for 60 seconds. Okay, so I will be watching for 60 seconds. Looks like 30 more. Just have to awkwardly stay here. Okay, so I will be putting this under the tray, the staining tray. And then with tap water, I'm going to rinse it off. And then I'm going to let the slide dry again. Okay, so I went ahead and I let my stained glass dry for a couple of minutes. And then I already went ahead and I put it under the microscope. So right now I have it under the 100th magnification. And as I'm looking and observing, so it's purple in color. And I actually see like a big patch. They're like long, it's like long. It looks really neat under the microscope, not gonna lie. So it's like this big patch. I don't know how to explain it, like long longitudinal circles. It's very interesting how this came up. So I went ahead and I also tried it with my high and my low power, and I noticed that I couldn't really see anything until I chose the 100 magnification, and with that one I can actually see a better picture. So I will go ahead and now take my picture of it to add it to my exercise homework. Okay, So I already went ahead and I labeled my next slide, which with 2 -M, and it says to cut open the Congo red. So I'm going to cut the tip of it, and I'm going to place a dot just like that. And then it says to scrape some cells from your mouth and inside of the cheek. And then I'm going to add it to the red Congo dye. And then it says to locate a blank slide and then pass it through just like this. And then it says to completely air dry before I can view it under the microscope. So I'm going to wait a few seconds there. Okay, so to me it looks completely dry, so I'm going to try to view it under the microscope and see what I find. It's hard for me to view it with the goggles. So I'm not really seeing much. I'm going to change the magnification and change it all the way to the 100 magnification and see if I see something different. So I'm kind of seeing like circles, small circles this time, not very big. Having a hard time focusing here. Okay, yeah, so I just see small circles, so that's my observation for the second slide. Okay, so this, I will be demonstrating what I'm going to do for exercise number 2. This is the differential stains. So I went ahead and I already labeled my 3 slides with the E. coli, S. cerevisiae, and S. epidermidis. So I have my inoculated loop here. So what I'm going to do is start with the first slide, which is E. coli. And I'm going to open this and sterilize the tube here. The tube with the candle light. And then carefully I'm going to grab my inoculated loop and carefully go inside the E. coli and grab a sample. I'm going to set the E. coli aside and put my sample on my slide in the middle of the circle that I made here on the slide. And now I will put the inoculated loop aside because I will be disposing in bleach. And I will be sterilizing the E. coli tube again. Okay, so now I can go ahead and close it with the cap. And I am going to let the E. coli air dry for a couple of minutes. So I will be repeating the same step for the next 2 slides, and I will check back. Okay, so now that I've let my slides air dry, I will go ahead and heat fix under the flame. So I will pass each slide over the flame 5 times. So I will start with the S. epidermidis. So I'm going to start backwards. Okay, so here I go. 1, 2, 3, 4, and 5. Now I will grab my next slide, the S. cerevisiae, and pass it over the flame 5 times. 1, 2, 3, 4, and 5. Now lastly, I will grab the E. coli slide. So the same, 1, 2, 3, 4, and 5. So I already went ahead and I put the crystal violet gram stain number 1 on 3 of my samples. And it's been sitting here for 60 seconds. So what I'm going to do now is add the E. coli onto my tray and then put it under the tap water. I think I did it a little too fast. Okay, so I am going to let my stain dry. Sorry, I'm going to let my stain dry for a little bit, and then I will be adding the gram stain number 2, which is the iodine, and I will be letting it sit on there for 60 seconds. So I'm going to just do that right now. So I'm going to add 1 drop. So now we're counting 60 seconds. It's really hard to record yourself and then try to do this with one hand. So I will now add it to my tray, and then we're going to rinse it off with tap water as well. Okay, so now we are adding the third dye, which is the Decolorizer Gram Stain, and it says number 3 on there. And this one will only be sitting on this on the slide for 30 seconds. Okay, so I added 1 drop. Now we're going to count down. Just making sure it's not staining the surface here. Okay, so now that it's been 30 seconds, again, rinse it off with tap water. And now I'm adding the last Saffron Gram Stain number 4. And as well, this is 30 seconds on the slide. Oh. I'm gonna add 1 more. I don't think I did it correctly. Okay. So counting down 30 seconds. Okay, so I'm gonna add this to the tray and rinse it off with tap water. Okay, so now that I've added all 4 gram stains to my slide, I'm going to let it air dry, and then I will be go ahead and doing the S. cerevisiae and the S. epidermidis. So I'll be right back. Okay, so finally, now that I have stained all 3 of my slides, I went ahead and I grabbed the E. coli slide and I put it under the microscope. So as I am observing, I have it under the is it the 4th? Sorry, yes, the 4th magnification. I can actually see with the E. coli like little like purple long dots. That's what I keep seeing as I focus on the slide. I'm gonna change the setting onto the magnification, sorry, to the 100th magnification and see if I see the same thing. I actually don't see anything with that magnification. So I am going to write my observations onto my exercise, and I will be doing the same for the S. epidermidis and the S. cerevisiae. And that has been all for this exercise. Thank you for watching.